Okay, so I'm just going to jump right into this then. So my name's Saif, and I'm here from Basecamp Research. Thanks a lot for having me. I'm a senior data engineer at Basecamp Research, and I'm going to talk to you about our particular use case, which is trying to create a sustainable metagenomic supply chain for the bioeconomy. Now, I'm just going to jump straight into, well, I work for Basecamp Research, and to tell you who we are. So essentially, we're a biotech company. And what we are trying to do, our main kind of fundamental mission, is to try and provide biodiversity guardians who are people with some kind of responsibility for some part of nature. So someone who was running a nature park, for example, or a government body in charge of the rainforest, just stakeholders in particular areas of biodiversity. And we're trying to offer them a fair and equitable way to essentially be able to utilize the resources that they have, because there's a lot of valuable resources in biodiversity. And we want to get what their resources through to the biotechnology innovators, which could be anyone. I mean, pharmaceuticals would be an obvious example, but across many, many sectors. So to just power something called the bioeconomy. So I'm just going to talk about what the bioeconomy is. So the bioeconomy is essentially just the economy trade, which is based on anything which is derived from biological organisms. And this is just a couple of quotes I pulled up just off the internet to just kind of demonstrate the potential value here. There's one from Forbes saying that the estimate here is like $4 trillion as a potential market value of the bioeconomy. And here's another one from the World Business Council for Sustainable Development saying around $7.7 trillion as a potential market value of the bioeconomy. So it is quite important for the people who have responsibility for the biodiversity to be able to actually access some of this, because it has no value unless it enters the marketplace. Otherwise, it's just there in the ground. Yeah, so just to kind of summarize the issue, if we've got two potential players here, right? So the person on the left is our biodiversity guardian. So what they're saying is, I have value here, and I could send it out into the world, but if I sent it out into the world in such a way that I lost control of it, it could be commercialized, someone else could make billions and billions of dollars, and wherever the actual IP came from, the value never ever flowed back there. But then on the other side, you got our biotechnology innovators who want molecules which are ethically sourced and performant. The ethically sourced thing is actually quite important because legislation is changing worldwide. At the UN, there's something called the Nagoya Protocol, where they're really trying and enforce the fact that all this kind of biodiversity data should be ethically sourced and traceable back to its point of origin. So this is where we come in. So one of our key abilities at Basecamp is we are capable of going around the world to collect these samples. So this is just an example of the kind of sampling area we've actually gone to. So this is Antarctica, where we've had a couple of samples, soil samples extracted from here and here to examine the biodiversity that's available in Antarctica. And that's one of our key selling points, that we have the ability to go out to these places. So our discovery process goes along these lines. So the most important first step is to actually acquire the permits. So we have a biodiversity team, and the biodiversity team engages with essentially your biodiversity guardians, but also at all levels. So national governments, state governments, municipal governments, all the way down to private landowners. You need to negotiate with all these people to try and get a permit. And that permit actually has to be informed consent. So they know what we're doing, they know what we're going to actually do with their data, and then they agree to it, and then they also work out what benefits they're going to acquire if we actually manage to commercialize something that we discover from this area. So then the next step, now the biodiversity team does their thing, that's crucial to anything that we do. And then our field team comes into play. So our field team is a bunch of people who are like field scientists, but also people who excel in survival skills. They can go to extreme weather conditions and they can thrive, essentially. So they go out to the sample, they go out to the space that the permit covers, the geographical geospatial area, and they sample it. Our samples are essentially soil samples, so they take soil samples, and then they also collect the metadata around that soil sample. So metadata could be anything like, what temperature was it? Was it raining that day? Just anything you can think of about a particular bit of land. We're going to collect all that metadata. Now, when we have these samples, the next step here is then to essentially you stick that soil in a blender or a shaker, you extract the DNA from the sample. And then when you have the DNA, we then sequence it. We either sequence it in-house or we use second-hand commercial partners. Now, when we have this data, we then have DNA sequence. DNA sequence is not the primary endpoint of our product, so it's actually protein. So revisiting high school biology, DNA genes, they code for these things called proteins, which are the kind of actual actors in this space. So yeah, so what is a protein? Essentially, it's a functional molecule. So anything that happens in life, any biochemical reaction, any structural aspect, it's all essentially mediated by proteins. So cellular machines, you could call them. So in data terms, what does it mean? So it's actually big text files with 20 permissible characters which correspond to the 20 amino acids that make up a particular protein. Or it could be a 3D coordinate of an atomic structure where someone's crystallized the protein and then they've fired X-rays through it. So you end up with something like that, which you can render as a picture. So that's literally what the actual molecule looks like. Okay, so proteins, incredibly valuable, we said, but it can be utilized almost anywhere in the sense that, so we go from cosmetics through to pharmaceuticals, alternate food, meat-free foods, biofuels, and so PCR, for example, is also driven by something exactly like this. So this is something that everyone's going to be very familiar with right now. Literally came out of a bacteria that was extracted from a hot spring. So that was a very, very valuable example of something that can be extracted. And then down here, also very exciting stuff, gene editing, biotechnology tools, treating diseases of the future. Okay, so this is the actual initial data challenge then, right? So it's like, how do you maintain this connection? So you want to go from a piece of paper, which is a legal permit, to a place on the ground, which is a geophysical place, to some bit of soil that you extracted from that place, keeping the metadata connected to that. And then once you've got that metadata, then the proteins you've extracted, and then whatever annotations you've done in that. So you've very quickly just gone from a piece of paper to millions and millions of data points. So this is our initial data challenge. So at this point, I'm going to try and essentially explain how we decided to take this on. So when I joined the company, we had, I think there was a very similar version of this slide at the keynote, but this was something we had to do. It was like, okay, well, how do we actually model this data? I came from a relational background. I spent a lot of time working with relational databases. So my first impulse was, yeah, well, I'll stick it all in tables and join it. But then we said, okay, there are other alternatives. So we looked at the other two, kind of NoSQL versus graph as well. So first off, we looked at document databases, but we ended up dismissing that clearly out of hand just because of the nested structure of our data. So if you've got a sample, and then inside a sample, you've got a protein, and then you've got all the annotations, it's just turning into this kind of crazy Russian doll structure, which will not be easily queryable. And then we looked at relational versus graph, because we still kept relational in the game. We did an empirical study, but then we also thought about it theoretically, and fundamentally, graph won out because any piece of annotation that you have on the molecule is patchy. So you might have 5 pieces of annotations on one molecule and none on another. So relational databases would not handle that very well, because you end up with a whole bunch of tables with a whole bunch of NAs in them, which is not particularly useful from querying or performance purposes. So just to kind of model our actual data flow then, we end up with our protein pipeline, which essentially runs the DNA through, and then we identify the proteins in there. We have the sample data flat files, which I mentioned earlier. We have curated data, which comes from our domain experts. So someone who's a specialist in a particular type of protein will actually go away and assemble some extra data, which we can then put into the graph. We then have public data. So a lot of this comes from like government databases. The NIH, for example, has lots of big protein databases, and also various European and Japanese equivalents. And then on top of that, we then have ML-derived insights. It's like, okay, so if we actually look at these proteins, can we look at what the ancestral sequences look like? Can we look at what potentially any future sequences could look like? Things like that. Any kind of insight that you can withdraw. And then all of these things are pushed into a large ETL data munging pipeline, and then driven into Neo4j, which is essentially where all our data lives. We have a massive graph. I think we're at something like 120 million proteins and counting, so 120 million nodes, and then we're up to around 1 billion relationships. And again, growing every day because our team is constantly out there and the data just keeps coming. Yeah, so just to kind of sum up, this was our supply chain as I just talked about. So this, once you actually have the graph in place, you can maintain this line. And now, because we are actually starting to sell these things to various customers, there is actually money coming in, and we can literally walk this line back now from the biotechnology innovator back to the protein, back to the experiment, back to the bio. And even if it's a small monetary value, you can then actually deliver that value down the chain. And that's what will actually make this process work and create this virtuous cycle to actually power the bioeconomy. And this is what we've actually ended up with now. So we have a knowledge graph, and within the knowledge graph, we have the metadata that we talked about, the environmental, geological, chemical conditions. And then the second layer, we now have the microecology. So we know what's in there, because when you look at the DNA, you can work out what organisms it came from, and you can look at the genomic context. So if you have a protein on a particular chunk of DNA and there's 5 proteins in one direction and 2 in the other, you've got a context, and they might all be coming together to form some kind of secondary product. And you only know that by being able to put these links together. And then similarly, we then have the ML insights that we talked about. And this is our primary IP at Basecamp, and this is how we deliver our value with the construction of this structure or knowledge graph. And now I'm just going to segue into a case study, which was actually very interesting for us. So a customer came to us with a very interesting problem. So if you're traditionally trying to work out what a protein does, you look at its ancestors, right? So you construct a tree. So it's exactly like a family tree here, except this is a molecule. And then you're saying, okay, I want a molecule that does X. Just give me all the molecules which are similar, and then it'll probably have function A. And then function B, all the molecules which are similar there in function B. But then if you look across on the right, the problem they actually gave us was it's just popping up sporadically on the tree. So in evolutionary terms, there's no reason for any protein to have this function or not have this function. So there's no obvious signal in just the primary amino acid sequence. So this was a particularly interesting problem for us. So it seemed like a very good use case to try and then apply the GDS library in the sense that we could say maybe there isn't signal in the primary similarity, but maybe there's signal in the kind of network topology of these things. So then essentially our approach here then was a very traditional machine learning approach, which we came up with public data, curated data, where we said, okay, we have a whole bunch of positive examples where the proteins do have the function. We have a whole bunch of negative examples where the proteins don't have the function. And that's the tricky thing to assemble, actually, because to say that a molecule definitely doesn't do something is complicated. But then having constructed this data set, we were then able to essentially pull out all the examples within our main graph of proteins that kind of could potentially have the function, and then apply essentially just Louvain, like community detection. And then if you see in the center there, we came up with these purple ones, which were a whole bunch of new potentials sequences for this particular function. And actually, these have been delivered to a customer now. And this is actually a very, very interesting example of kind of in silico detection of protein function, because ultimately, we didn't do anything in the lab. Well, we sequenced the DNA, but we didn't go off and express the proteins. We didn't compare them to anything. So this is a step change for moving forward in biotechnology, in my opinion. And then what was more exciting about that, then we took the graph approach, and then we went in a completely different direction. We looked at a graph convolutional network, and we looked at protein structure, and we are essentially completely orthogonal to this, because those two approaches are involving structures, and this is only sequence. So, but literally every one of the hits that we looked at we got a kind of 3-way agreement on this, and that was very exciting because there's no obvious reason for this to work, but it does. So we have to think about why it does. So it was very, very exciting for us. And I'm just going to leave that there and open up to questions if anyone has any.